will be the scientific methods with which we do science, and especially with which we do biology. Biology, like the other natural sciences, it has a core steps in its scientific methods, which is the experiment, and it has one of the core tools of it, which is the microscope, apart from the other characteristics of the scientific methods which is existing in other field of sciences. There are many ways of listing the scientific steps, but I prefer this list of scientific methods which starts with raising questions or formulating questions. Actually, the question formulation comes from a reasonable observation or a reasonable information that you have from whatever you have, from reading, whatever, and from this you may come up with certain questions, and raising the question, stating the question, identifying the question by itself could be considered as the first step. Then what follows is the data collections, that you try to collect data or data so that to answer the question that you've raised, or the question that you're given with, or the problem that you're facing. Now, your data collections is not necessarily has to be strictly to answer the question, but all the areas that are needfully or necessarily has to be an answer to the question. You're not going to just collect all the jacks of information that are not necessary for your what we call problem to answer. Then the next thing is, from the data collected, you're going to extract out those which are possible answers. I mean, they're going to tell you the reason why this situation happens, and you're going to get the answer for your question from the collected data. You have the data collected based on answering the question. You're not just simply collecting you're not simply collecting junks that are not going to be useful for your proposed question or for your stated question. Now, from the data that you've already collected, you're going to extract a possible answer, a scientific guess. And those possible answers in scientific guess are not necessarily to be one or two, they could be more. And that answers, we call them hypotheses. Now, you have the hypothesis. From the hypothesis, even you're going to use your reasoning so that you can come up with the strongest hypothesis. You can use inductive reasoning, you can use deductive reasoning, you can use according to the nature of your question. You can use your large amount of data to get the information, or you may use some general statement that will help you support what you have selected. So, using those reasonings, you're going to come up with hypothesis. And from the hypothesis, you again use those reasonings to choose one of the strongest. Then you take the strongest, and when you take the strongest, you're going to apply that to be tested. This time, the hypothesis is going to be called prediction, which is nothing but the strongest hypothesis. Now, this strongest hypothesis is going to be tested. And the testing process of the prediction is called experiment. And we have the experiment in what we call biology. And the experiment in biology is necessarily has to involve certain requirements like the chemistry and physics. The requirement in the requirement of the biological experiment, as far as it is dealing with living things under a controlled environment, like for instance in laboratory, then you're supposed to have two groups, which are called the control group and the what we call experimental group. For whatever you're doing with the experimental group of a specific variable, you're going to have an equated control group with all the necessary things in place except for what is going to be testable. Now, this is what we mean by the control and the experimental group. We're looking for a result in experimental group. Whatever we find in experimental group, it is going to be translated. It could be interpreted, or the result could be verified or falsified based on the control group result, which is the control group outcome. And these are going to just serve as an evaluation parameter, whereby we're going to say due to this and due to that these things happen and that things happen. We'll just do some examples in order to just introduce that when we talk about the experiment later. Result is whatever you gain from the experiment, whatever the outcome of the experiment, we call it result. And after result, we have our interpretation and our conclusions. We try to interpret that result, and from the interpretation and the result, we're going to have our conclusions. And this conclusion may lead us to verify the prediction, so that we can come up with theory after a repeated experiment done by the very person who's doing this and by the other people, the other persons. Or you can have a kind of a loop forming towards the hypothesis if the prediction is found false or the statement is falsified. This is one way of dealing with scientific methods. Another way of dealing with scientific methods is that you, like I said earlier, make an observation. From the observation, then you're going to come up with certain interesting questions, and those questions have necessarily to be answered, and you try to answer these questions, and what your answers are going to be hypotheses, and out of these hypotheses that are supposed answers, supposed possible answers that you have, you're going to select one which is the strongest, which is prediction, and you're going to test that prediction by experiment, and afterwards it becomes a kind of a general guidelines or a kind of a general theory, or it could be falsified, and again you'll be back forming the loop towards the what we call the hypothesis. Even the general theory format, the theory that's being produced, is going to again be questionable from different observations. That's why theories are changing after a long period of time or a short period of time. So still the loop is formed long after the theory is postulated or formed. So we have observation followed by question, and then an answer to the question, hypothesis, and the strongest one, which is the prediction, put into test experiment, and then that experiment bring about a theory, which is a result, and followed by theory, and that theory is going after a long period of time will be questions or observation, and big cycle is there. The small cycle is that from the prediction, which is going to be tested by the experiment, a falsifying statement or falsified conclusion will bring about those predictions, selections back to hypothesis. So from hypothesis to prediction, experiment, conclusion, prediction, experiment, conclusion, hypothesis, prediction, experiment, conclusion, hypothesis. That's another loop that will be formed. So the falsifying and the verifying statement will bring about either to form an immediate loop or to form a theory, so that it will stay longer before it's changed if it is subjected to. Another way, which could be scientific methods forming a cycle, you have for instance observation, hypothesis. Like I said, that hypothesis will be tested, you're jumping the prediction, no problem. Maybe you have only one hypothesis, and then that will be analyzed, interpreted, concluded, and everything, and then the results are again will be questioned, and then again there will be kind of fast loophole format. One way or another, we are going to follow the very first one, which is the most common one. Going to the experiment, which is the core staple of scientific method, is experiment, as a matter of fact. And when we talk about the experiment in biology, like I said earlier, there is a set of two groups, experimental group and the control group. When the experiment is over, there will be a need to report the outcome with a proper format reporting. Why do we need the experimental group? Why do we need the control group is one question. When you have the control group, whatever the result that you get from the experimental group, it could be used in order to verify or falsify the statements, the outcomes of the experimental group. For example, you may be dealing with certain organisms, okay, for the deficiency disease or for the infectious disease of a certain food that it brings about, or the contamination of certain food, we can say. Now, say for example, we can use we can choose, say for example, deficiency disease study, vitamin deficiency disease study. So we have deficiency disease. For example, we have some lab rats and we're looking for deficiency disease of X vitamin or an X mineral or an X food. You have certain groups of organisms, groups of lab rats, for example, and with everything that is needed, you're going to put them into one group, and you're going to deprive them with a variable. You're going to deprive them the variable, which is the vitamin X, and you're going to you're going to have another group with equity with the other one that's equally having everything and also the variable which is being tested. Now you have two groups that are having everything, one is not having only the vitamin X. Now when you compare those two, this one are supposed to be a reference to the outcome of this one, which is the experimental group. Now in the experimental group, what you have is you're looking for the outcome of the deficiency disease of the X. And if, for example, there are diseases that occur here, and the symptoms and the disease, the nature are exactly the same with what will happen to these organisms which are having that vitamins, then for sure the reason is not the vitamin. But if those organisms with the deficiency of the vitamin X, form or display certain symptoms and certain problems of the deficiency disease, while the others are not, then that will lead us that the cause is, due to the deficiency disease of vitamin X. The interpretation of the results needs a control group, and the results is expected in the experimental group. So we need the experimental group for this reason. The formats of the report of any biological experiment could differ from one way or another, but mostly the format starts with a date, and then you have the topic and subtopics of your experiment. Then you have the objectives of the experiment. After objectives, you have the apparatus. After the apparatus, you have the procedures. And after procedures, you have the results, you have the interpretation of the results, and the outcomes of the results, which is what we call conclusions.